Well, good morning, everyone. Welcome aboard research vessel Falkor. I'm Dr. Brendan Brooke. I'm a marine geologist and chief scientist on this survey that's being run by the Smith Ocean Institute. You're actually viewing live footage from the remotely operated vehicle SuBastian down at 500 metres depth. Today's dive is on northern Flinders Reef. It's about 250 kilometres east of Cairns, Australia, and we're in the Coral Sea Marine Park. So our research cruise at the moment is investigating the geology, geomorphology and ecology of the deep margin of a selection of reefs in the Coral Sea Marine Park. So these reefs are very unique and they're very little described, especially the deeper realm of the reefs that we're looking at. So this is down the mostly very steep margins of the reef. So the research team aboard, and also we have members of the research team based ashore that are joining us remotely, just as you are. And we're viewing in super high resolution the seabed via the SuBastian's amazing cameras. So the idea today is that we're going to observe and also collect some geological and biological samples as we rise slowly from around 500 metres depth up to around 100 metres on the upper margin of North Flinders Reef. So what I might do now is hand over to our coral expert, Jeremy Horowitz, who will be able to describe some of the amazing features you're currently viewing on the seabed of North Flinders Reef. Thanks, Jeremy.
Hello all, my name is Jeremy Horowitz. I'm a PhD student at James Cook University. My affiliations are with the Museum of Tropical Queensland and the Centre of Excellence for Coral Reef Studies. And I'm on the ship because I'm interested in black corals. I am studying the evolutionary history and the taxonomy of the group. And so, yeah, we're here for 3 weeks and hopefully we'll show you a lot of cool stuff. So what we have right in front of us, I'll zoom in, seems to be, at first thought, I thought it was a black coral, but it looks like a soft coral. Looking closely, and then going to a focused view, you could see the polyps seem to have more than 6 tentacles, which would indicate it being an octocoral. It's interesting. And so we're interested in collecting black corals, soft corals, sponges, and yeah, we're going to go ahead and show you our first biological collection. We're going to collect a fragment of this coral. And while it's being collected, you can see a lot of associate invertebrates on this coral. So this coral is providing home to a pretty diverse assemblage of species. And so what they're doing is getting off the ground, so they're getting some elevation, which allows them to feed on passive nutrients going through the water. And it's a little bit safer for them than down on the seafloor. Yeah. I also like to introduce our ROV pilots, Ben and Chris, who are excellent, and they're going to expertly collect all the biology and the rocks and the sediment samples seemingly with ease.
Jeremy, how long of a sample do you want? 2 inches, 3 inches?
3 inches. 3 inches will do. Yeah. So why we're collecting biology on this trip is probably a good question to answer. So my colleagues and I are using an advanced molecular technique called ultra-conserved elements. And so what we're going to do is take these samples. We're going to look at the morphology, which will allow us to identify the species based on morphological features. And then we're also going to extract DNA and sequence that DNA with UCEs, which will give us an indication of how it's evolved, its evolutionary history. And based on those two prongs of information, we're going to be able to identify the species and update the taxonomy of these various coral groups. So we have experts for the hard corals and the soft corals, and I'll be working on the black corals and also the sponges.
What's that? We don't have a jar,
Where's the depth?
Oh, you want to?
99.1, just on the top upper left.
That matters. Let's do a floater or sinker. It's floater sinker.
It'll probably be neutrally buoyant.
Okay.
Probably needs a lid.
Yeah, we'll definitely have a lid on it. It's just whether or not it'll be escape while we're doing this.
Okay. No, it won't float.
Okay.
No, it won't float up. It should drop.
Jeremy, just so it's easier for you to reference, this screen is what we'll be broadcasting on at the lunch that's left. So.
All right, so we've collected our soft coral sample and it's now going in the bio box. So in about 6 hours from now, when the ROV is back on the ship, we will subsample the tissue in 100% ethanol, and that'll be from a fragment, and then the rest of this larger fragment will be put in a bag and sent to our experts for identification.
Before you go, you just want to try to snap into the sea bottom a little bit. Let's see how easily you can get.
We see a sea anemone over here in front of us in orange. So what we're going to do now is try to take a sediment sample. We're just testing to see how soft the floor is.
Hold on, I'll help you out a little bit. It looks pretty hard, so I would have doubts whether I'm going to be able to get a good sample, but we could definitely take some of the topsoil with our suction samples.
Okay.
Yeah, that's right.
Cody, which one did the
I measure 1 through 4.
1 through 4? All right.
I'll get you a sheet.
All right, so 2. All right, it's in jar 1, so you're going to go ahead and grab your suction sample. Super bright. Parse out a little bit if you need to. Unfortunately, just touch. Tell me when you're happy. That's all right. Give you your seat as well.
Yep, I'll look at that side. That's the 5 feet from the microphone.
All right.
Jeremy, you want to tell us what you want to
That's the screen.
sample?
on YouTube.
You want a general area where you want?
Marty.
Where you are right now is fine.
Good there? Okay.
That's cool.
We're gonna go into one.
Is anywhere here?
Yeah, anywhere there,
that's
like soft stuff and tingly stuff. Great, thank you.
Just one surface, or we'll dig in?
Dig in a little bit.
So we're going to be taking some sediment samples on all the dives that we do, and our geologists are going to look at the sediment samples after, help them understand how this
Great,
habitat
yeah.
has
That's
been
funny.
created.
Alright. Alright, now we're in 2, so that's not gonna go anywhere.
Storage location, you tell that's called in New Zealand.
Should be jar number 1 or suction sampler 1.
Slurp.
Yep, that'll work. Yep.
So everyone on YouTube and Facebook, feel free to send questions your way, and I'll try to keep an eye on the questions as they come in.
Camera's yours and we'll put it.
All right, we're going to start our transect.
Okay, so I'll head over to the ROV start.
Yeah. So we collected this coral because we landed right in front of it, but the first thing we're going to do this morning is do a 500-meter transect. And we're going to do these transects on every dive, and this will allow us to compare the assemblages at the different reefs.
I can do that.
Hey, Gerald, we can look up top. I'm gonna have the.
Nudie, we won't be collecting fish during this trip. We will just be collecting geological samples and coral and sponge samples.
There we go.
Cody, full blast up.
Yeah, we won't be collecting fish on any of these dives while we're out in the Coral Sea.
There he is.
I got it. Figured it out.
So just to reiterate, we're at Flinders Reef North, out in the Coral Sea. This is actually the first time coral collections are being done in this part of the Coral Sea, which is really exciting. We really don't know much about what lives in the Coral Sea, although a lot of these corals look like they belong to certain species found elsewhere. It's likely that a lot of these are similar looking but different species. So that's why we'll be collecting and trying to figure out all the species that live in the area.
Wow.
Cool fish.
It's amazing fish.
There's a picture.
Okay, cool.
Trying to see if I can view
The way that it works.
Beautiful
is
fish.
If this is the shits
Yeah.
from the world,
You're right.
and you can control it and do what you want, and then outfly around after this and the other ones.
The size of the eyes.
Awesome. Yeah, so we have a fish here.
Some more.
That's as much of an ID as I'll give you. But there is a pretty large following of fish people. So I assume as they come on, they'll be able to identify different fish.
Mardi Gras fish.
Mardi Gras does look like a Mardi Gras fish. The purple and
A green eye is what people are saying. So we'll go with green eye fish.
Got total spotted green eye fish.
Start point. You happy with the pitch?
Sure. Yeah. So we'll begin the transect? Oh, we're heading towards...
Yeah, right over there now.
Okay. So we're about to head towards the start of our transect.
So, can you swap the command of the sit for the coordinate about? Or is that?
That might throw it off.
Do we want to try and get over off?
I know where you.
Get out?
I'll work on it tonight. I'll get on deck.
Okay.
Or a bit of what's photos?
I
Yeah.
think they're thinking of getting a rock before we start the transect.
You know what?
We're going to try to pick up a rock before...
Yeah, just before we start the transect.
Want one of these
we get
rocks?
A rock, yeah.
So we're going to grab a rock before we start our transect.
Big rock, little rock? I guess point on a rock. Big rock. Like the one we got yesterday would be great.
I will
Yeah, about a
over to you all.
About a fist size or
fist size or 2 fistfuls.
Rock right in front of us, so you can pick that up. Look at my sit, buddy. Think you need that? Is that too much for you? Too much for you? We try to break off the bigger rock.
hey, what's that? Big fish.
Oh yeah, there's some big fish in the background.
You want to go look at it first, or you want to go look at the fish and then come back to the rock? Or you want to look at the rock?
Oh, we can grab the rock. Yeah.
That's rock. There's a small one right there in front of me. You can see it?
Yep. Outer cats.
Any options right here then?
Sorry. Yeah.
Hey, Chris,
Yes, sir.
remember doing the transects with Rachel?
Yep.
So we want that same setup with the.
So the same
Camera,
height, same angle.
Yep. So you remember it was like 35-degree angle with the camera, outer cats off. I think all cat things off. And that's it.
Go.
So while they're grabbing a rock, I'll look at all the corals in the area. So we have here a black coral. The genus is Stichopathes. We'll be finding a lot of these whip black corals during these dives, and we'll certainly collect many of them.
This one right below here.
Our primary interest in the whip black corals is that it seems like, although the whip in the shallow and the whip black corals in the deep look identical, based on preliminary molecular analyses, they're different. Actually, the deep ones fall into an entirely different family, which is odd. So it seems like convergent evolution's at play here, where this unbranched morphology just seems to be favorable at different habitats. So we'll, during these dives, collect both shallow and deep whip black corals and sequence them and see how they're related and probably describe an entire new genus.
Would you like to split the rock?
And just one of the smaller bioboxes.
The same one as the coral, or that?
Can be. We also see an anemone here. We could do a little zoom in action.
Do you need to? Do you want a video of collecting the rock, or do you want to just do it?
Yeah, we could video clip. Yeah, we can
Yeah?
show it.
All right.
Yeah, I'll just take over. All right, let's grab a rock.
The big one? All right. I don't know if you got it in you, but it's kind of old.
Nice guy, John.
I don't think you can do it. Try to get like that little feathered lip at the top. That's that indentation, so you keep that. There you go. Good. Good job, Denver. Alright, portion out. Do you want portion out even that's a look at the overview of the rock? Or are you gonna keep it in frame?
Oh, there was a shrimp behind it, was there? Yeah.
Looks like a rock to me. Well, what's interesting actually is when we collect these rocks, often you find lots of little invertebrates living on the rocks. Actually, last year on the ROV Investigator with Rov Pilots, who was narrating the last cruise, we were collecting lots of rocks, and we ended up finding a new species of carnivorous carnivorous sponge, and it's about 5 millimeters in height and very intricate. So, yeah, let's do a little quick search, a little scan. But when it gets back on board, we'll do a good search for anything living on the rocks, and we'll subsample it for later identification. Oh, there's a little crustacean. It's gonna join us for the ride. And I think the things growing on the bottom, they might be bryozoans, I'm not sure.
Seems to be a lot of worm tubes in there, Jeremy.
Yeah, lots of worm tubes. Little microhabitat for lots of different species. Oh, I'm excited.
Good first step. All right, then.
All right. We're gonna drop this rock in the bio box.
There's a coral in there, so
And which one?
so you open it slowly. You want it in the same box.
You want it in the same box as the other with the coral? Or you-
So, Kat, we're hopefully gonna use all the samples we collect to identify the species and document the biodiversity in the Coral Sea, which is generally just unknown. Like I was saying before, although a lot of these corals look like things found elsewhere, given the distance from where the other specimens were collected from, it's likely that they've speciated and evolved and therefore are different species. So the only way to identify the species and document the biodiversity of the area is to get a sample, morphologically identify it, and also molecularly. When we bring them back up to the surface, so they'll be brought up in the cold water, similar to the temperature now, which is 10 degrees Celsius. And once we get it on land, we will quickly bring it into the wet lab and fix them in ethanol and preserve them. Can we? We're done with the rock?
Yes, sir.
Okay. Can we grab?
Yeah, go.
So we're gonna try to grab one of these black corals while we're here. Looking at this one.
Right up on your screen, clear good. See the lasers? The one in between those lasers.
So, yeah, the specimen we're collecting.
That one?
Yeah.
Do you want to take a couple of at first?
Yeah, sure. We'll zoom in quickly. All right, so you can see, so this is the genus Stichopathes, and the species is really hard to tell. You identify the species by looking at skeletal spines. So there are the spines, the skeleton of the black coral has these spines that are repeated along the colony. And so what we do is we strip the tissue off of the skeleton, off of the colony, and using a scanning electron microscope, look at these small skeletal features, which are generally less than a millimeter in height. But you can see here, this is Stitchopathes, and I know that because it has one row of polyps. If it had multiple rows of polyps, it'd be a genus called Sauropathes. But again, we know very little about the relationships between these whip black corals, and it's likely that we're going to have to break up these genera into multiple genera and redescribe lots of the species. So we'll go ahead and grab the sample.
Same box. Is it floater
No. But you can put it in the same box. And can you take the whole colony?
or full colony? Which one do you want first? Is it left or right? Yeah.
The left one.
All right. Then the base. So the base and then
Hybridization, we have no idea, but actually the molecular method that we're using, ultraconserved elements, has the ability to detect hybridization. So that's one of the many things that we're going to be looking at when we finally have these specimens sequenced. Yep. So the reason why we're going to take the full colony and not just a fragment is because for these whip black corals, because they have so few morphological features, we have to look at how the skeletal spines vary along the length of the corallum or the colony. And it does tend to vary between the top, middle, and bottom. So in order to identify this specimen morphologically, we're going to take a fragment of the top, a fragment of the middle, and a fragment of the bottom, look at the skeleton of each section, and that will tell us morphologically what species it is. And of course, we'll sequence it as well.
All right, let's go back here for it.
Jeremy, can you tell the camera that you're collecting a sample and what it is?
All right, once again, we're collecting a black coral. The genus is Stitchopathes. And we're collecting the full colony, and we're going to use this coral to identify the species and include it in our ever-growing phylogenetic tree to understand how it's evolved.
Box one, put open. Oh, sorry, one or two. One? All right. Huh? That's two. You want two?
Yes.
Okay. Very right here.
Yeah, so the goal is to collect what we call morphotypes of each, what we think is a species. So we're going to try not to collect too many of one species, but it is important to collect a few of the same species because although they may look the same, they probably aren't all the same species. But we're going to limit our collecting per site, per species, to a few morphotypes. Yeah, so the DNA is going to be a second line of evidence that we use to identify the species. So, morphologically, we may call it one thing, but recent advances in molecular work allow us now to really understand how different specimens are related. So we're going to use both pieces of evidence in order to identify the species, which will result in different identifications than if we were to just use morphological IDs, because morphology can be misleading.
Still on you a little bit. Your dog's touch more. Still caught up on there, but you gotta pick up. Sticking out. Come on, fall. go too far. Might need to push it in the bottom corner. You see it?
Regarding collecting, if you can imagine, how much of this coral sea we're surveying, it's 0.00001%. And so my point is there's a lot of them out there, and we're certainly not going to be eradicating species through these collections, but the few specimens that we do collect are going to provide us with a lot of information in order to conserve biodiversity of these habitats. That's how we, the only way to know how to protect different habitats is to understand what lives in these different areas and how far the different species found in a given area extend to. So collecting these now allow us to better conserve these species and these habitats in the future. And with regards to the coral sea and the Great Barrier Reef, the marine park that was established many years back, it was created without biodiversity data, which means just large areas were protected from fishing and drilling while other places were open. But it wasn't based on biological data, so it wasn't as effective as it will be now when we have this information about what lives where and how rare it is or how common it is. And so that will allow later in order for us to design the protected areas to maximize our conservation impact. All right, so now that we've done that exciting collection, we'll start our transect, and we're going to try to minimize stopping and collecting, try our best. So we have a continuous 500-meter transect to compare between sites, but no promises. This is technically our first dive where we're collecting, so I might get excited and want to collect one or two things.
We always try, but it never seems to work out that way. We always try to keep going, but it never seems to work out that way.
Yeah, that won't be very helpful. Yeah, it's actually amazing that we're on this ship now and doing this survey. It was uncertain for a while, of course, because of the pandemic, but we were able to get all the permission and everyone's been checked. So everyone on the ship is COVID-free, and yeah, we're extremely happy to be doing this survey, which has been planned for a very long time. And it's a very important one. It's the first, as I said, it's the first time we're collecting in this part of the Coral Sea. So it's going to provide a lot of new information about this marine habitat.
Before we start this, we might have a bit more success taking the core. Let's try it here.
Okay.
I'll
Core?
Yeah, right.
Yeah, I'll go ahead and
All right.
I'll come over here where it looks the sand here.
Okay.
Thanks, Chris.
So, yep, although we just said we were going to start our transect, we of course are changing our minds. We're going to take a core sample first, and then we'll do our transect.
Try it.
We should. Yeah, we should. We did. There were some holes last time. Actually, was on board. And before we had to set up, we'll look up straight up. This was up down. We'll just right along the road and stay that far.
It was probably about 130 metres lower than today.
What does it say?
The question's now.
Stephen, to answer your question, the last ice age, how far down was sea level? About 125 meters. So a lot of what we're going to look at during these transects and these surveys, especially as we get a little bit higher, is what used to be on land. Yeah, Stephen, and we're also mapping these habitats to get higher resolution maps in the Coral Sea. So there has been some mapping done, but during this trip we're going to be filling in gaps and upping that resolution in lots of sites that have been surveyed and mapped.
One or two or three or four or five or six.
Mention the sediment.
That was one.
Number one, yep.
Yep, so we're about to do our first sediment, our first core. So on this ship, we have lots of people with very different specialties. So I'll be passing off the mic at times when there are other people better suited to answer your questions or explain why we're doing what we're doing.
Anywhere.
Yeah, I think so. Yeah, hi everyone. It's Brendan Brooke back talking to you from RV Falkor, where we're looking at video footage, live video footage from the remote operated vehicle SuBastian, and SuBastian is currently using its robotic arm to collect a push core of sediment in water depths of about 500 metres on the northern margin of North Flinders Reef in the Coral Sea.
I'm not getting it. A little bit. Keep going, keep going, go down, go down, go down. You're pushing down on the thing and holding the copy. You see that? See if you got it. See if you got it. Doesn't look like it's catching. Bring it all the way up. Got a half one maybe?
Yeah, that looks good.
Hurry up. Look.
Woo! So,
This is that. It's pushing the, and that's gone. It's gone.
yeah.
I would try. Clear it out for you. I would try it again, but
Yeah, we're just having a bit of trouble collecting the sediment.
you push the handle.
And I'm pushing from the up and up.
Okay.
Here we go. We're going to have another go at trying to collect some sediment from this area of sand. You can see some very nicely formed sediment ripples indicating the bottom currents here are strong enough to at least at times mobilize the sediment.
So I'm seeing that spring release at all. We'll try.
Yeah, we're just trying to make sure we get a bit of a vacuum seal to hold the sediment in before it gets pulled up.
So yeah, I'm gonna try a different one.
As you can see, we're having a bit of trouble with this core, so we might try one of the other core barrels. See if that can give us a better seal so that sediment stays in the tube. It's very free-flowing sediment, so it's quite difficult to retain it in the tube as it gets pulled up, unless you've got a very good seal at the top that creates a bit of a vacuum to hold the sediment in.
Alright. Alright. Oh, you're going in. Go ahead, sorry. Aft a little bit. Still do one, keep going aft. You're sick in. There you go. Alright, stand by. There you go. Alright, calm down. Upward, just touch. There you go. Forward, 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 down, forward, down, forward, down. Alright. You're on the edge. Well, there you go. There you go. Drop it.
Beautiful.
Nice. Hello.
So we're going to have another go at collecting a push core of sediment here on the northern margin of Flinders Reef in the Coral Sea Marine Park.
I'll stop that. Just trying to settle you down. There you go.
Our pilots are trying to manipulate the core barrel into the right position, and hopefully this time the top of the barrel seals the sediment in a bit better.
Let's see. Back go for it. Come down so we can see. Look like a bunny. Come on. Inboard. 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 Come inboard. Not back. Inboard. There you go. Rotate. Rotate. Go down.
Wow, great job, guys. That was not easy. So Chris and Ben, our expert ROV pilots, are here today.
Alright. Okay, so I'll stop. Stop. Success. Go back at it.
So this quite fine to coarse sand is biologically produced calcium carbonate sediment that's been shed off the reef, which is about 500 meters above us from this point.
And you're allowed 1/3 of the coat today.
Yeah.
1/3
So
of the coat.
this material's been produced by organisms that produce calcium carbonate sediment, such as halimeda, encrusting algae,
Out all the way.
corals, a whole range of organisms.
What height do you want to start at? 1.7 meters. Off the seabed.
Now we're about to recommence our transect up this very steep side of North Flinders Reef.
So Dr. Brandon is asking what altitude you want it, how high do you want it off the ground?
Yeah, put it about 2 meters above the seafloor if possible,
Alright.
at about 0.3 but not.
1.3. Alright, what lights you want? What were we doing?
Huh?
What lights were we doing?
It's like
Chris,
annotation.
if you want to get stationary,
Get up.
bring your camera down just a little bit, center it up, bring it down.
So the pilots are now setting up SuBastian with the appropriate angle on the cameras and lighting level and distance above the seabed before continuing the transect up this northern margin of North Flinders Reef in the Coral Sea Marine Park. So we're about 250 kilometres east of Cairns in 500 metres of water, and you're joining us live from research vessel Falkor.
The lasers on or off?
It's on. It's
Alright, lasers are on.
Come left a little bit, Chris. Right there. How's that look? Alright, Chris, that's it.
Alright, I'm gonna push. Yeah.
So, outer cats, mids, inner cats on.
Aight.
It says it's not getting that was
Yeah. Gotcha. Are you
1.9
now?
1.9.
I changed this more. Oh wait, hold on. That's an issue.
Sure. Oh yeah.
also an use those.
Is that better there?
Okay, Ben.
Alright.
Sure.
So you're watching live video from the remotely operated vehicle SuBastian. It's 500 metres below research vessel Falkor, the Schmidt Ocean Institute's research vessel, and we're conducting a deep water dive from a depth of about 500 metres up the side of North Flinders Reef in the Coral Sea Marine Park.
You guys ready? Alright.
Bridge ROV.
You ready over there?
So as you can see, it's mostly sediment-dominated seabed here, so the seabed's quite mobile. Sediment's being mobilised by the bottom currents experienced in this part of the reef margin.
Thanks, Eric.
So you ready?
You're ready.
You told me to wait.
I didn't tell you wait.
Okay. All right.
So
We're
go.
doing. Okay, we're going.
Okay, so transects.
Start transit.
Starting.
Okay, we're off on our 500-metre transect, during which we'll maintain a consistent attitude towards the seabed, so that we've got a consistent area of coverage by SuBastian's cameras. We'll maintain a constant speed and also consistent levels of lighting. By doing these standard dives, we can compare data between dives.
Ask yourself.
We're now at a depth of about 492 metres, and we're still passing over a sediment-dominated seabed. We've got carbonate sand here, carbonate sand and gravel, and it's strongly rippled, indicating the bottom currents here are more than capable of mobilising this sediment, so moving it mostly to the west in this position.
Where did you teach at university, or did you independent work, or?
I'm at Good morning, everyone. Just to remind you, we're streaming live from Research Vessel Falkor. You're viewing live video feed from the remotely operated vehicle SuBastian, which sits about 500 metres below ROV Falkor, and we're slowly climbing up the northern margin of North Flinders Reef in the Coral Sea Marine Park. And welcome to everyone that's joining us live through various social media outlets. And hi to Rob Beeman. I can see you've joined us, Rob. And of course, Rob led the previous survey in the Coral Sea Marine Park that's recently finished. That's the Visioning the Coral Sea survey. There's lots of information on that survey on the Schmidt Ocean Institute's website, where there's a very nice web page on Rob's survey. Likewise, on that website, you can also see the web page for this current survey, which is titled Seamounts, Canyons and Reefs of the Coral Sea. At the moment, we're in water depth of approximately 480 metres, and we're passing over a sediment-dominated seabed. It's very strongly rippled in places. Carbonate sediment, calcium carbonate, that's been biologically produced, largely up on the shallower areas of the reef, and it's now being mobilised and moving down the reef margin.
The young guy, what's his name?
Jackson.
Jack, the tall young guy.
Yeah, Jackson. Is he here?
Just a quick question. When we get now you get that footage now inset where you can actually see where on the reef we are. We haven't done that yet.
Ah,
In terms
yes.
of location, we'll do that.
Yeah.
Yeah.
Sound right for trade?
So that's where we are.
Great.
And do you want to talk about how we did the transects and all that stuff?
Yeah, that's a good idea. So once again, welcome everyone. You're joining us live from research vessel Falkor. You're viewing live video feed from the remotely operated vehicle SuBastian. That's tethered to Falkor in a water depth at the moment of about 475 metres. We're passing over a sediment-dominated seabed ranging from gravel to medium to fine carbonate sand. We're following a 500-metre transect up the margin at the northern end of Flinders Reef. To explain to you in a little bit more detail the importance of this transect, I'll hand over to Dr. Andrew Carroll, and he can explain the great advantages by applying a standardised approach to these sorts of transects. Thanks very much, Andy. Over to you.
Thanks for that introduction, Brendan. So it might appear that we're sort of not looking at a lot here, but one of the important things that we're trying to achieve across all of the sampling we're doing across the entire marine jurisdiction is bringing away with us some quantitative information about the seafloor. And in order to do this, we need to standardise our methods. One of the ways we've done that is through a series of field manuals that has been developed for a number of platforms such as ROVs, AUVs, towed video transects, etc. So it's fantastic when we come down and see these unexplored parts of the seafloor and we're able to sample and describe some of the organisms we're seeing, but it's also important that we undertake a standardized portion of video above the seafloor that we're able to then quantify later on. So this portion of seafloor might contain a lot of animals, a lot of benthic organisms. It might also be sand, like we're seeing at the moment, or different raised features, geological features that can provide important habitat. So using a platform called Squiddle Plus, we're able to take screen captures. So every half second we're taking a screen capture of the seafloor, and what we can do post survey is analyse that information in order to be able to make comments about the percentage cover of certain benthic organisms occupying these areas. We do that consistently among transects, and that gives us a point from which to compare these habitats. So sand for the moment, but I'm sure we'll see something sometime soon. This is the other one.
Okay, this is Jeremy Horowitz, and we'll try to identify some of the biology that we see during this transect. And then once the transect's over, we'll explore, look for some interesting critters to collect.
Jeremy, where are you from?
So, I'm from the States. I'm from New York.
They told you so.
From Long Island.
Long Island?
Yeah.
Really? My roommate's from Long
So
Island.
New York City.
Oh, yeah?
almost, yeah.
Outside the city. Long Island City, outside there. But I've been in Australia for about 5 years. I did my master's at James Cook University and my PhD, which I'm nearly finishing.
I said, Joe mentioned that I think it sounds like Ringo.
Great. Yeah.
So Australia just more in line with what you wanted to do as far as study and whatnot.
Yeah. Well, the university was, their research is top notch. They have researchers, head of their fields. So I started my PhD after visiting the Museum of Tropical Queensland, and I was interested in black corals at the time. And so when I first arrived in Australia, I met up with Tom Bridge, who was the curator, and asked him if he had any black corals. And he said, yeah, I've got a lot of them. No one's ever identified or looked at them before. So I went into the collections and identified all the species and published a paper on what actually was a collection of black corals from the Coral Sea. So there was an expedition called the Sedaris expedition about, I think, 30 years back.
Okay.
And so a ton of biology was collected through dredges, and certain groups were looked at. You know, if there was a taxonomist with that particular interest, he looked at the corals that were collected within his interests. Otherwise, they were just kind of put aside. So we went through about 50 black corals, and it ended up representing range expansions for 7 species, and we suggested to synonymize 2 species. And yeah, it was one of the first papers to understand black corals and, you know, what species of black corals live in the Coral Sea.
Okay. Were you watching the last one we did?
Yeah.
I think that your work was mentioned
Yeah.
during the dives. Some of the things that you had done. 'Cause that's,
Yeah.
That sounds like a familiar source. I think that you're being bragged on.
Yeah, Rob Beeman did a very good job narrating the last trip. Hopefully we can similarly entertain you. But Rob will be on the second leg of this trip, so I'm sure everyone will be happy to hear his voice again and have him talk to you about the Coral Sea and all the research that he's undertaking.
I do it from here instead of just scare around his house.
Yeah, it's a small group of researchers on the ship. There are 8 science team?
9.
9. And yeah, with diverse skill sets. So we have people I'm interested in the corals, people interested in the geology. We're also going to be running AUV dives to map a lot of these areas.
I am following the transect line, correct? The green line.
Yeah.
Sorry, just to make sure before I make a turn.
And the universities are all within Australia. And of course, there were lots of challenges getting on this ship. So a lot of people who had meant to be on this trip weren't able to enter the country. So that allowed Australian researchers, people nearby where the ship left from, which is Cairns in Queensland, to join. So it's a smaller crew, but enthusiastic and excited, very excited.
Yeah, and we've got Dougal remotely.
Yeah, and we have lots of experts working remotely, although they can't be on the ship, working with us, all day to identify dive sites and what to collect and all the logistics. Yeah, looks like maybe a sponge. We'll have plenty of opportunities to collect. Seems like there's this one similar looking critter popping up. So once the transect's over, we can grab one of those, figure out what it is. Yeah, so the dives, we're going to try to do dives as often as we can. Looks like some, maybe a soft coral. It's hard to say. Again, I don't want to stop mid-transect. Could be a black coral. Hard to say. Yeah, not many fish, maybe a few. And actually we saw a nautilus at the very beginning of this dive, literally seconds after we went live, or before we went live, but it escaped before we could pan to it. But hopefully we'll see some nautilus like we did on the last expedition. Yeah, correct, oak tree. We saw a lot. And the plural for nautilus? What's the plural for nautilus?
Moving their nautiluses because that's from the Greek base. So nautilus is not no lives.
All right, nautilus is.
What's a collective noun?
I guess we can make it up.
We came up with some ideas. What was our collective?
Of course, it was.
What do we call a group of nautilus? Any ideas?
Chamber of nautilus.
Chamber?
That was one of the ones that were suggested last session.
All right,
Yeah.
okay.
Chamber.
So hey, transcends cruise. I think we go with it.
There we go. Buffet, not bad. feet afloat. All right. We're gonna have to narrow that down. But we have 3 weeks to figure it out. These things are interesting. So we're at 140 feet, meters, sorry. And how far into the transect are we?
An error for measurement,
Probably
250.
no, it's more than that now. 300 now.
All right, so we're
About here's about 500.
okay. So we're over halfway through our
Yes,
transects.
over halfway.
Then we could zoom in on some cool critters. Discover what's living down here.
Great job, updated your position.
Update, thank you.
I see a fish. Any ideas? Ah, the fish is out of screen. It came and it went.
So it appears that our angle is kind of steeper. So it's going to slightly change the angle that we're viewing. I can come up a little bit to try to maintain it, or I can stay at the 2 meters. Whatever you prefer.
Whatever you guys think. The angle's changing. Do you want them to go up a little bit?
Angle's different.
Yeah, so it's coming up a little bit steeper. So I can maybe come off the seabed.
Socially distancing on the boat. Yeah, it's a bit challenging. But Falkor made sure to do as much as they could, so we haven't filled out all the rooms. So there's space between people during sleeping. And everyone was tested before we got on the boat, which makes us fairly confident that we're all COVID-free. But we do have precautions in case someone gets sick. So Falkor's doing everything they can to make sure this trip is safe for all the people aboard.
Keep that angle.
Anemone. Oh. Interesting looking coral. Probably gonna come back and look at that when this transect's done. I see a black coral, a whip black coral. Without looking closely, it's hard to identify what genus it'd be. Some more corals. Yeah, we'll probably come back to this area in a few minutes.
John, can you do it instead of the top right corner?
We're gonna, in a second, show you a map of the reef we're surveying, give you a bit of perspective.
I'm getting meters.
100 meters to go?
Yeah, once you pass this, you got, give it 20 meters.
Alright, so what we can do is we'll go a little beyond the 500 and show you the overlap, so like 510, 520.
We,
Jeremy, you said you're from Long Island? We'll do a little,
From where to where?
yeah. Just have to find Long Island.
It's the long one.
it's that it. That's it. The whole thing.
So you got any questions from Long Island, Jeremy?
Huh?
Any questions from Long Island?
Yeah, anyone here listening from Long Island? It's, I think pretty late at night or early in the morning, so I expect not.
Just shy. Just after 15 years.
Texas? Connecticut. All right. Wicked. Is that from Salem? Just kidding. So,
About 50, 60 meters left, Jeremy, just to make sure.
all right, the transect is nearing its end. We're almost there. So, on this expedition, so we'll be, as I said, we'll be collecting hard corals, black corals, soft corals, and sponges. So we have Merrick Ekins, who's collaborating with us on this project, and he'll be receiving all the sponges that we collect. Hopefully some glass sponges and carnivorous sponges, and hard corals. We have Tom Bridge, who will be chiming in from time to time when we get up into the shallower mesophotic depths.
Bridge, you have a new position.
And soft corals, we have Kathy McFadden, who's going to be helping us with our identifications and our molecular work. She's also using ultra-conserved elements, and so is Tom Bridge. So this trip will provide a whole lot of new insight to the various anthozoan groups. And yeah, in a minute or two, we'll be able to slow down and observe and zoom in and sure, look at some fish as well. So we're seeing this, I think it's a sponge. pretty much the only thing we're seeing large enough to see from this altitude. It's another sponge.
Yeah.
And then in there for 2 days. Same song. Same hook of the same song.
I only know one of these songs.
You only know one Eminem song. What hook for you? You're my roommate. I don't know. Oh, yeah, sorry. He's a fellow Detroiter. Michigan.
Apparently, the ROV drivers are going to sing Eminem. A little rap.
See, I don't know if it's my head. It's one of the chords. We don't want to drive away or listen. How many we got online watching?
Quite a few.
Any questions for us, the crew, while we end this transect?
We're just gonna overlap a little bit journey
All right.
and just go a little bit past the top.
Yeah. We won't go beyond 200,000 meters, 2,000 meters, right? For these dives.
Yeah, we shortened it a bit.
For all, like for the trip.
Yeah,
generally.
2,000 meters in horizontal distance maximum.
Okay. Yeah. So yeah, our dives will be generally from a 2,000 meters going up to mesophotic depths, as shallow as we can go. What sort of training do the ROV arm operators have?
By and large, it's on the job. There are some schools you can go to, do basic ROV theory and whatnot in application, but it's pretty much you observe for a while, and then you're allowed to do small things in very controlled environments. With someone sitting right next to you in case you get to a bad spot. And eventually you just become more and more comfortable, more proficient, and then there you go.
Yeah.
I think Ben's been operating ROVs since the beginning of time.
Since they first came out.
Yeah, you know, it's still there. Through the Shilling's the one who built the arms that are on the vehicle right now. They do have some courses you can take. You get a little bit of opportunity to use the arms there, but it's basically just come out here and get told what to do. Practice until you're decent enough that they let you do it on your own.
It's pretty cool.
That's just about 500 meter right there, but so we'll go a little bit beyond just to make sure we have full coverage for the transect.
All right.
Good time to it's a good time to do anything. No, I wasn't zooming anywhere you were. That's 510 meters or something.
Oh, so we're good with the transect. All right, transect completed. Good job, everyone. Yeah, we have cameras looking at all sorts of angles. So the lasers are on now, and they represent 10 centimeters. And so now we're just going to look for a good place to settle down. I guess any of these areas to look at one of these things. Yeah. Is it a sponge? Let's see. Let's explore.
Sit down and look, or you just want to fly over?
Yeah, let's sit down and take a nice image and a sample. Oh yeah, all right. Oh, there's a fish looking at us.
It's not happy.
It's not happy. We're yeah. He's trying to tell us that we should maintain social distance. Rapping about peduncles. Don't hold your breath on that. All right, I'm going to try to zoom in on the sponge, see what we're dealing with here. All right, so it's a sponge. Oh, and there's a little critter. At the base of the sponge. And can we zoom in more?
Is it a shrimp or a crab?
Looks like a crab. I'll try to focus for you. There we go. Yeah, a crab of sorts. And you can see a brittle star actually behind the sponge. Which is pretty cool. So why don't we grab this sponge while we're here?
Okay. Where would you like it?
You can go in the same bio box.
Okay. I'll zoom out just to see where we are to make sure that it's
Yep.
successful.
Yep, so
Make sure you can reach it.
let's say it's
Oh, yeah, it's easy.
15 centimeters
It's
in
about, well,
height.
let's see. Yeah. The angle.
All right.
Can't tell.
So we're going to grab this sponge and give it to our sponge expert, Merrick Ekins, who I'm sure is very excited.
We are at the end of the transect, so we're going to explore some, so we won't be heading due south anymore, but it's going to come down there and stand by. We'll figure out our next move.
Alright, coming just up there.
Outstanding. Thank you, Eric.
Yeah. So we can do more sampling now.
Remember,
I think they're still looking.
Alright, okay, we're
Right, you ready? Am I going
ready.
Ready?
So, all right, we're going to collect this sponge, and what Merrick will do to identify the sponge, sponges have what are called spicules, which are these little angled spongin particles that have distinct characteristics. And so what he'll do is he'll take a fragment of the sponge, remove everything else, and then take close-up images of these spicules. And that will help him identify it based on morphological features. Yeah, looks like a glass sponge, I think. I'm not a sponge expert. A lot of these glass sponges are undescribed, so the chances of collecting something that is new to science is pretty high.
Alright, let's start portion up then. Roger.
Can you zoom out, Johnny?
One or two, which would be easier for you? Two? Alright.
Can you shake it a little bit to get the sediment?
Yeah.
Yeah, give it a little shake.
Good look at the
Yeah, looks pretty good.
Alright, follow you over. That's a good start right there. Nice look at it, get some good pictures.
Thank you.
So we first found glass sponges on our last expedition on the ROV Investigator, which was this time last year. And Rob had talked about this story during the last Falkor expedition, but however, he forgot to mention one thing. When we first found this cable, this intertwined cable of what ended up being a glass sponge, we were at first thinking that, oh no, we collected internet cables or some sort of cable laid by humans because it was so perfect and cylindrical and spiral. So I think I had about an hour of debating, trying to figure out what it is, looking at the literature. And then I called Rob Beeman over. He took one look at it, pulled the piece off, and took a bite and goes, it's silica, glass sponge. So, geologists are good breed.
It's not advisable to put sponges in your mouth, though.
Sure, as a general rule, we don't eat our biology that we collect.
Especially some sponges are quite toxic.
Yeah. Yeah, so we're not advising that you eat sponges. Some could be toxic.
Disclaimer.
All right, the sponge is being dropped into the bio box with our soft coral and black coral.
Good collection down here.
Eat your study organism, yeah, I've heard that as well. I haven't done it for black corals, and I definitely don't plan on it. Okay, so we're gonna do a bit of exploring. See what else is living in this habitat on this reef.
Alright.
Yep.
Camera's yours.
Yep, we have permits for collecting corals, octocorals, hexacorals, and sponges, and also rocks and sediment, and plankton as well. All right, let's look around a bit. Oops. Yeah, maybe we could stop here for a second and see if it looks different. Sure. Actually, yeah, why don't we collect? All right, we're gonna stop in front of these sponges, which seem to be different than the one we just collected. Actually, there seems to be two different sponges here.
Yeah, zoom out on my system. Zoom out.
Yeah, we'll try to get Merrick Ekins to speak a little bit later during one of these later dives to explain about his work with sponges. It's really interesting. All right, we're just getting situated.
No, it's just that I'm on an angle, so I can't sit down straight, so I'm gonna spin. See if I can get a little bit flatter positioning. Set that up. All the way up. Yeah, big and in, of course. That's what it is. Try a different angle.
Yep. We're just on top of the sponge now.
Maybe the left side of the vehicle is higher. Sediment, so it takes some concentration.
Okay, so these sponges seem to be each about less than 10 centimeters. So I think we'll grab the one on each end. So the one to the left and the one to the right. But first, I guess we'll take a zoom in. Okay, so let's look at this guy. That's interesting. Oh, there's a little crab. Why don't we say hello to him for a second? Hello. All right. So you can see the design of the sponges. You can see all the intricate designs, the spicules. So let's look at this other one. Oops. All right. Focus on this. All right, we're going to try to focus on the smaller sponge for you. And you can see the substrate. Looks like shells, lots of broken up shells that have accumulated here.
That's a little bit better. about as much as I can do, really.
Yeah, okay. All right, so we're gonna collect these 2 glass sponges.
All right.
Okay, I'll zoom out and we can go ahead and grab them. Okay, it's all yours.
The little one on the right.
Yep. Alright.
Yeah.
You see how we made it?
Do I see it? Yeah. These little grey flakes look like they're running.
Yep. So Marty confirms Halimeda. Okay, first of 2 sponges being picked up.
Bring it up for a video.
Okay. All right, we're gonna just take a close look at it before we drop it in the box. So you can see it's got an attachment to the substrate. Okay. Is it possible to focus on that? I'm gonna try to get a zoom in on this sponge. There we go. Yeah, this is not going to be squishy. This will be a hard sponge made of pure silica.
Let's see, since that's the only picture, I'll do it better. There he goes.
There it is. Oh, and there's a little critter inside who's coming along for the ride. So it's actually interesting to always, if possible, to collect the associated species with the coral that we're collecting, because it'll provide us insight into what these corals host. Lots of these corals will host lots of high diversity of associated fauna. So yeah.
All right.
Yep, let's throw it in the box.
Go back. Whoa. Hold on. A little premature there. Behind that. Fortune out slowly. We are on a hill, so let's push some of this up. Fortunately. How's that for you, Ben? That's good. What's going on now? That was weird. I wasn't doing anything. Yeah, that was fine. I'll go back. Watch out some more. Alright. Interesting. Pushing you forward. I don't know what's going on, to be honest with you. I'll rather slow this down a little bit. I know what I'll do. Gotta give you enough space to work, and then I'll figure that out there. How about yeah. Because I moved this around a little bit, I might have to reposition to get that done. I was going to grab it down. I got faith that I'm pretty good. Alright. Right above it.
Yeah, so we're up on a slope, so pushing out the bio boxes dug us into the ground a bit. But that's alright.
There you go.
We're getting a pretty good collection of life here. Yeah. Let's grab the one to the left.
Smaller?
Yeah.
Alright. Yeah, and new ones. After one hitch, though, we showed our love by scraping them up, banging them up, and then spent weeks rehabbing them just as we had it for a month.
If it's easier to get to the one to the right, that's okay. Okay.
It's got ones left.
Yeah. Got some pros here. It looks kind of looks squishy. We'll have to look at it when it gets back on board.
Connected. Might want to come in separate too. Looks sticky as well, so.
Good question, Dave. Yeah, if we find loose invertebrates, we're obviously not going to, we might not know where it came from, but we do have the video to refer to. But if we have a specimen where we're especially interested in its associates, we can put it in a separate box.
Get a picture of it.
You don't want the debris, right?
So if there's anything on it, it's just for us to know if there's associates. Yeah, right. You're good.
Okay, I'll stop.
Oh, look at that. It's got some maybe some fossil hard corals, maybe some hydroids. And then, so you can see the spicules hanging off the bottom. Those are the fin. We could probably zoom in a little more. That's
Very
it.
closer as well.
Yeah. Yeah. Why not? We could try to look at those long strands of the spicules.
Can you come any closer?
I got trees. Yeah. Locked in.
We'll try to give you as good of a view as possible.
You want to zoom out a little bit?
Yeah. Look at that. All right. We could see some forams on here as well.
Thank you guys. Wonderful.
Yeah, beautiful. And you could see here, this is a spicule. I don't know if I could zoom in and we focus on so they're not all straight. They'll have some sort of structure. So I'm going to try to focus in on this spicule that seems to
It's gonna focus.
grow in 3 dimensions. Yep. That's good. Yep, so you can see how that one branches perpendicular to the primary spicule. It seems to have maybe 6 spicules going out radial. But I'm no sponge expert, so we'll leave that to Merrick. And just look up a little. Anything else? Oh yeah, lots of different types of spicules. More forams, corals. All right, let's throw it in the bio box.
Which one was that going to?
The one above.
Where did
Right.
Biobox 2?
Yes.
moving on. Just a hair and shop.
What are you gonna do then? Candy.
There you go, Ben.
Yeah, we were very excited to be able to get on board and head out to the Coral Sea. There was, of course, a lot of uncertainty surrounding this trip. So yeah, we're thrilled. The stuff we're collecting now and the stuff we'll collect over the next 3 weeks will give taxonomists and phylogeneticists work to do for the next 5 years. All right.
Continue on moving, transect.
Yeah, we'll just let's look for some sort of a rock habitat that we can find some corals attached to.
Are we still doing the transect or?
No.
Okay. Cool. Well, I think I see something to the left. So it's
Yeah. This one sponge is a bit upset, which took away its friends.
Soon. Soon. You're all both on with that now? Is that what you're saying? You're better material? I know you're going to do that. Just do it. Tell me, seriously. You're tickling me. You want to take a look at that there, Jeremy?
No.
Let's keep moving up. You want to find a big
Something with. Yeah. I won't be too picky though.
Okay. Something a little more likely to it.
Yeah.
Get it.
Something with polyps.
Sounds good to me. That's what we got there.
Yeah, so we have Dougal on YouTube, who will help you answer your questions. Unfortunately, he can't be on this expedition, but we are going to collect the data he wants for him. Hopefully, as he expects, some good video footage of some. As we're descending, we'll take video of jellies swimming by, but we'll let Dougal explain his research at a later date.
Okay, you are. I just don't go around Pollard by Phil. So. I don't see nothing much on that, Jeremy.
No, not much. Oh, you know what? Why don't we take a look? Why not?
I'll land out, let you know when.
I see maybe is that a cup coral on top there? It's very big. Maybe an anemone. Okay. Oh yeah, I see a. Well, this could turn out. This could be a good rock. What do we got here? Oh. All right. Not this. All right. So the dive is nearing its end. So let's see. What do we got here? Sorry for the short, abrupt end to our first dive.
Picture that.
That's interesting. Oh, I see a cup coral. Why don't we grab this cup coral At least then we've gotten a little of each group. Yeah, so I'll just try to zoom in a little bit.
Do you want to get with the suction?
You think that'll work?
Yeah.
All right.
Oh, wait.
It's all yours.
All right.
So we're going to collect this a zooxanthellae cup coral. So a zooxanthellae means it has no zooxanthellae, doesn't require sunlight to grow, and it's a hard coral. And it's solitary, meaning it just consists of one polyp. And you tend to find these in the shallows inside caves, or basically anywhere in the deep. And as with many of the other coral groups, very little is known about their evolutionary history. So this sample will tell us a little bit about what it is and what it's related to. Sorry to end the dive early, but our AUV has to get in the water, and we can't have both going in at the same time. We actually started this dive a little bit later than expected, so our future dives will be longer and allow us to do more exploration in this otherwise unknown habitat. Okay, we're gonna suck. And I think we got it.
I wanted to suck up this rubble in case it's
Yep.
stuck in there. I think it went in, but
Yeah, might as well. We'll call it Sediment Sample 2.
We're grabbing that cup coral real quick. Did you get it? I think it fell out.
I think it went in real quick for the rest. So, yeah, thank you all for watching along. There'll be plenty of dives over the next 3 weeks, so please tune in. This was dive number 371, Expedition FK200802 on RV Falkor with ROV SuBastian.
Alright.
just down there, Geoscience.
Oh, sorry.
My name is Jeremy Horowitz. And regarding a tag, we'll have to get back to you. We want to make sure we give you the right Twitter tag.
It's hashtag vision and quality.
Get a nice fly.
Yeah, it's hashtag vision and coral sea, one word.
Thank you, buddy.